So let's move to Ruud. Ruud, presentation, please. Ruud, the floor is yours.
Programs that other people don't have. Okay, so I'm working in Wageningen Plant Research, which is part of Wageningen University and Research. And as François already mentioned, I'm studying, among other things, plant development, specifically tomato fruit development in ripening. And we started using CRISPR-Cas mutagenesis in tomato about 7 years ago. But first, about gene editing or genome editing in crops. So editing in crops started way before the invention of CRISPR-Cas mutagenesis. In the mid-90s, other nucleases were used, like zinc finger nucleases. We have, from 1999, the oligonucleotide-directed mutagenesis, which one American company is using and commercializing. In 2009, tail nucleases were introduced, and the company Calixed is commercializing products from that. So actually, the two products that are now on the market that are gene edited are using these technologies. In 2012, as you well know, CRISPR-Cas was discovered, and in 2013, this was first applied in plants. So probably like in other organisms, we see three main applications of gene editing in plant breeding, which are called SDN1, 2, and 3 for site-directed nuclease, to include also the older nucleases. And SDN1 is causing small deletions or insertions that interrupt the function of genes, with which you can delete unwanted traits in plants. And you could see this as replacing chemical or radiation mutagenesis that has been used already for tens of years. SDN2 is the substitution of one gene version, an allele, for another one by making a limited number of changes. So that could, for example, include base editing or prime editing. And it resembles the backcrossing for integration of traits from one cultivar or one variety into an elite variety, which takes a couple of generations to achieve. And with gene editing, this is much faster. SDN3 finally is the insertion or replacement of entire genes, including replacing with genes from other species. So then it starts to resemble transgenesis somewhat, or from sexually compatible species. So advantages for breeding in crops is speed, precision, and relatively low cost. But I should make a disclaimer that many things that are claimed that we will be able to do very soon are quite challenging for CRISPR-Cas. For example, I've been working with insect-resistant crops that contain a gene from a bacterium, Bacillus thuringiensis, and it will be difficult to achieve something like that with simple SDN1 or SDN2. So what could gene editing in crops be used for, or is it already being used for? That is, for example, the removal of gluten allergens in cereals like wheat. That's being done in our own institute, but also in Granada in Spain. Improving seed oil quality and make it healthier or better to cook. And Calixed soybean oil is commercialized right now in the US. Then there is not the best example, I think, but there is herbicide resistance, and Cibus, the company in the US, has produced an herbicide-resistant canola and is commercializing it right now. Maybe a better application is deleting susceptibility genes that a pathogen needs to be able to infect or invade a plant, and if you knock them out or change them, then the plants become resistant to pathogen infection. And the USDA has just approved a blight-resistant rice variety that was produced with CRISPR-Cas. You could improve the agronomy of existing crops, like flowering time, the stature of the plant, or the yield. Reduce food waste, for example, anti-browning in mushrooms or apple. Increase the shelf life so that you don't have to throw away produce. Mitigate some of the effects of climate change, like heat, salt, drought stress tolerance, or de novo domestication of wild plants or crop relatives, like producing new crops that already contain stress resistance instead of trying to change existing crops to make them more resistant. This is one of my favorite applications, and one example from my own field, not my work, is the domestication of tomato. Cultivated tomato was domesticated over several thousand years from a wild relative, Solanum pimpinellifolium, shown on the left, to produce cultivated tomato, Solanum lycopersicum, which is not so stress tolerant, but it is optimized obviously for horticulture. Like in some other crops, we know fairly well the major genes that were involved in domestication and that give modern tomato their properties. So researchers did CRISPR-Cas mutagenesis in Solanum pimpinellifolium for 5 to 6 genes in several groups, and in that way produced a de novo domesticated tomato, demonstrating that CRISPR-Cas mutagenesis allows you to do something like this in only a few generations. So the development of crop gene editing poses a problem for the perception by the public, the regulation by the EU, and the enforcement of those regulations. So current EU regulations stem from 2001 for the release into the environment, and they did not foresee such fast new developments. Also, our understanding of what is natural and of the plasticity of plant genomes has grown tremendously as more genomes are being sequenced. And we realize that also in nature, a lot of things change in sometimes very bizarre ways. Many of the edits in crops will also be indistinguishable from natural variation or from changes made through exempted mutagenesis methods like radiation and chemicals. So a lot of my colleagues and myself expected that the European Commission would treat these edited crops with small edits like the exempted mutagenesis methods. Unfortunately, the European Court of Justice decided differently. So it ruled that products from targeted mutagenesis like CRISPR-Cas or ODM are still regulated under Directive 2001 for the deliberate release of GMOs, while products that are derived from undirected mutagenesis, which cause many more mutations, unknown mutations, much larger effects, they are considered GMO, but they are exempt from regulation because they have been safely used for already years. And CRISPR-Cas doesn't have that claim. Enforcement will be challenging because of small changes that are difficult to detect. The Commission has no intention to change the directive, although many scientists argue for that. And the European Council of Countries requested a study from the Commission about the status that is due early 2021. So we're very anxious to see what they will tell us. In the rest of the world, regulation is either process-based, as in the EU. So if you do genetic modification, it falls under the regulations, or product-based, where actual product is being studied and its properties. So new developments in gene editing in crops, some have already been mentioned. New Cas nucleases like Cas12a are already being used in plants. Base editors are being used and they work efficiently in plants. Prime editing is just starting. So replacing small stretch of nucleotides is just proof of concept. And template-guided repair, homology-directed repair, is still very challenging in plants. So now switch to the cost action plant ad. The cost action plant ad is being financed by the Cost Foundation, and it is all about plant genome editing and assessing the full innovation potential and impact as one of the objectives. set the direction of future research, to promote a link between research and innovation in a socially responsible manner, and to examine the synergistic interactions with closely related fields like genome sequencing. Some numbers. The chairperson and the grant holder is Swedish University of Agricultural Sciences, and the chairperson is Dennis Eriksson, who asked me to give this presentation. We started last year. We already have a lot of participants from all European countries, but also neighboring countries and further afield internationally in the rest of the world. And it includes various stakeholder groups. The action has 5 working groups. Working group 1, technical platforms, is the one where I am the vice chair. There's also impact, policies and regulations, perceptions, and an overall spanning communication working group. We had our first meeting in Serbia, Novi Sad, in November last year. We were supposed to have another one in September of this year, but for obvious reasons that didn't go through. Hopefully next year we will have the second meeting. The action is involved in some of the consultations that are being held by the European Union and EFSA. And it organizes training schools. There was one about regulatory options in January 2020. There was supposed to be another one in March, April of this year about genome sequencing. But for the same reasons that didn't go through. So hopefully next year. A cost action finances collaboration, but not direct research. However, it does finance short scientific missions where a member researcher can go to another lab in the consortium in another country and stay there for up to 3 months to do research or to learn things. And the action is active on a number of social platforms like YouTube and Twitter. We have a website, and we welcome proposals for collaborations and joint events. Thank you.
So thank you very much. So we can start the question.
You can stop sharing now, Ruud, and Elena can reactivate the video.
Okay, so there is a question for Elena from Veronica Popa. How can you be sure that the impact of this strategy will only be local? And how will you mitigate the impact on the trophic chain?
Yes, actually, this is one of the questions that I think that this doesn't have answer yet, because as I tried to illustrate in the talk, there are evidences that mosquitoes can travel and can go, and there are no frontiers for them to go from one country to the other. And also in terms of the fact that there are more than 30 species of mosquitoes that transmit malaria, then the solution is, okay, should we kill them all? Because as the question puts the emphasis, they also play an important role in the ecosystem and the ecosystem chain. So this is not the solution either, to kill all the mosquitoes of the world, of all the species. So this is something that we will have to be addressed. One of the recommendations, the current recommendations, is first to deploy regional regulations, regional and international regulation for this type of gene drive systems that apply to mosquitoes. And because this is not that local. And the second is that always a gene drive has to have a B option, like either a reversal drive, so you can stop the process anytime that you need, or a system that is like self-exhausting. So after a number of generations, it will disappear by itself, like the daisy chain option that has been published recently and so on. But this is not being investigated yet in the case of mosquitoes.
Okay, so I don't see another question on the YouTube chat. So I have a question for you. So since the very beginning of this meeting, we all agreed that public engagement is the only way to make GE of genome editing to develop well and to be accepted by the population. So, as you mentioned in your presentation, there was a lot of discussion in Burkina Faso. There are a lot of ONGs like Terre en Vie who are arguing that the consent was not provided properly, as you mentioned clearly. But I wonder how much of the Target Malaria project is dedicated to educational program or to community engagement, because there are a lot of money spent for scientific development, but how about social development?
Yes, this is a very good question. If you go into the page of Target Malaria, they put a lot of emphasis on the efforts that they are making to communicate to the general public and to have this consent and this acceptance. But the reality is a bit different. So in the field and in the places, there is a lot of opinion again. So I think that the problem is the following: that this project is led by foreign institutions and is being implemented by local researchers, right? So up to which point the information is being appropriately delivered, or there is a good program in place taking place, I'm not sure about it. So it's among the objectives, but I'm not sure that it's being implemented in the dimension that it needs to be. Like, okay, because it's this Bana district, but and the project says that they are educating people about mosquitoes and about engineered mosquitoes, but this is very local. It's not a big, an ambitious project to a real and effective engagement strategy. This is my opinion.
Okay, if I can follow on that, we've learned from Ebola crisis that the presence of anthropologists or social science experts is crucial in the way that to facilitate the implementation of new therapy or new behavior. How about Target Malaria? Do you have social science project funded through Target Malaria?
As far as I know, no. So this community engagement is led by the local people that are implementing the project there, but the same scientists, but that are explaining the research and are giving talks and seminars and so on. But there is no like a specialist, like journalist or a campaign by radio or television or involving social organizations that help to transmit the message.
Hi, Elena. This is Luis. Perhaps the thing is whether you have to train the trainer. So whether you are training these locals in the way that they could deliver the corresponding messages to the local population.
I mean, really, you should take look at the lesson learned from this Ebola crisis, definitively.
Yeah, I think that it's something that scientists don't have. We don't have the capacity to arrive to that level of communication. I like to understand the social, the political dimensions, the tempo, when and how long, and which are the target. For example, in Burkina, villages are organized in the way that there is a chief of the village, and all the people follow that person. So it is very important to understand how the communities are organized and how they function in order to deliver adequately the message. If the project doesn't have the acceptance of this chief of the village, the whole locality, the whole population will be against, for example.
Okay, thank you. So there is a question for Ruud, always from Veronica Popa. How is an unwanted trait defined, and how is the impact of its modification being measured on a long-term ecological level?
Yeah, sorry, unwanted
Trait.
Unwanted trait. An unwanted trait is one that is well, how to say that, is amenable to improvement for agriculture. So just to give an example, wild grasses from which cereals were domesticated, they very easily lose their seeds because that is profitable for their spread. But it's not profitable for the farmer. So farmers during domestication have selected variants that do no longer fall from the plant and can be harvested and then be trashed later. So that is, in terms of agriculture, is an unwanted trait. The loss of seeds is an unwanted trait. This is, of course, strictly from an agricultural point of view, not from a biological point of view. And also you have to remember that agriculture is not really ecology. So considerations for agricultural plants are not the same as for nature preservation or for ecology. So I know this is a very big discussion, and a lot of groups are active against GMOs, and recently GMOs are being called gene editing is being called GMO 2.0. So the same kind of discussions will probably come, but you have to remember that there is already a lot of variation in nature and also in agricultural varieties and cultivars that we hardly know and which are much more invasive, much bigger than the small changes that you make with gene editing.
Thank you. Jennifer, do you have a question? Can you? Someone has to activate her microphone, please.
Thanks.
Yeah.
I have a question for Ruud. It's probably naive. I'm quite sure it is. But have there been studies on consumers of these products, these tomatoes, for example, who have consumed them? Have there been studies on their organisms?
Have there been
on people who eat these tomatoes?
In fact, there are a lot of different opinions in Europe and around the world. So
that I understand, yes.
Food products made with gene editing, they are not, well, except for the soybean oil, not much is on the market right now. But if you, for example, in Europe, interview people, as you probably know, it depends a lot on the kind of questions that you ask. If you ask, do you want genetically modified food where DNA has been changed? The large majority says no. But if you ask them, do you want a food, a tomato or something else that tastes better, is better for your health, protects against heart problems, is better for the environment, and so on, then you get a different answer. So it's still very much under debate. It's genetically modified, or in the future maybe gene edited foods are unpopular in some parts of Europe, but that differs. In the US, that is much less an issue with the majority of people, I believe.
Yes. Thank you.
Thank you very much for both of you, and we'll go now to the next session. Thanks.